Sweet Synthesis by
Emma Carr,
David Wasserman,
Jordan Saibon.
and Parsa.
DNA synthesis allows genetic information to be passed in mitosis and meiosis. It is semi-conservative and it occurs during the S phase and interphase. The S phase is when DNA replicates.
Since this is something that is difficult to picture, we decided to show this process by building a DNA strand using candy, because it gives you a visual to clearly show the movement that is happening. It also is able to show the different structures that are being used in DNA replication, and you can clearly see the different processes that are happening.
a reason why DNA synthesis is so important is because if the DNA does not replicate accurately, mutations can occur. In our research, we discovered an article titled "Cohesion between sister chromatids must be established during DNA replication." These researchers focused on the effects that can come from DNA synthesis going wrong, such as the strength of the bonds between sister chromatids.
Now we're going to demonstrate the process of DNA synthesis by using a candy model. We'll be using licorice as the DNA strands, a lifesaver as topoisomerase, NERS as the RNA primer and the building strands, milk does as a primase, the helicase will be a Sour Patch Kid, and a skittle will be the single-strand binding proteins.
First, the polypeptide topoisomerase binds around the strand as helicase comes and unwinds the helix. Topoisomerase helps the strands keep from breaking.
Next, the enzyme helicase comes through and starts to unwind the helix, which reads 3 to 5.
Now the single-strand binding proteins binds to the strands that were separated by the helicase and helps them not to stick together.
Primase comes and binds to the leading strand and starts laying down RNA primer, building 5 to 3.
DNA polymerase 3 starts laying down DNA.
Primase comes and binds to the lagging strand, as well as building the opposite way in parts, so it reads 3 to 5.
DNA polymerase 3 comes through again and builds on the RNA primer on the lagging strand.
The building of the strands lays the groundwork for the next steps of DNA synthesis, translation and transcription. Thank you.